Good evening to all. I myself, Nandini C., PhD scholar at CTCRA, Trivandrum. My presentation topic on genetic diversity analysis in indigenous and exotic and wild species of cassava using SSR markers. Cassava or Manihot esculenta is one of the most tropical root crops cultivated extensively in Africa, Asia and Latin America. It serves as staple food for billions of people and is valued for its ability to grow under environmental conditions like high drought stress, high temperature and low soil fertility. Cassava is also used for food, and also it used as food for animal, and in its industrial purpose is produce starch, sago, etc. Its origin is from Brazil and South America, belongs to Euphorbiaceae family. They are heterozygous and monoecious and protogyny in nature. In India, it's cultivated in Kerala, Tamil Nadu, Andhra Pradesh and Maharashtra. Cassava germplasm consists of indigenous varieties, exotic varieties and mild relatives. Indigenous varieties are locally adapted land races by farmers over generation. Exotic varieties or released varieties are introduced genotypes developed or collected from other geographical regions. Wild species represent the ancestral relatives of cultivated cassava and possess valuable traits such as resistance to disease, pest, and environmental stresses. Genetic diversity is essential for long-term survival and improvement of crop species. Enhancing cassava productivity, resilience relies on clear understanding on its genetic diversity. Traditional morphological characterization has its limitation, so making molecular markers such as SSR and SNP is precise for genotype identification and genetic diversity studies. Then methodology. To evaluate the genetic diversity, I used 26 cassava genotypes from CTCRA germplasm collections. For exotic collections, 7 are 13 indigenous collections and 6 wild species are selected. These are the table shows the cultivars I used in the study. Then fresh and young leaf samples are collected and DNA isolated by using CTAB method. Then isolated DNA's qualities and quantities were checked, then followed by PCR amplification. Then I used 10 SSR primers for this study. Then the PCR products are separated by using 2% agarose and photograph the gel using by gel document system. These are the PCR conditions. These are the list of SSR primers are used in study. After that, band scoring obtained from the gel results and prepare a binary data matrix, the presence of absence of the bands. Then the score data of individual SSR markers were used for the calculating the percentage of polymorphism and also polymorphic information content. After that, similarity matrix calculation by using NTSC SYS PC software. And this similarity matrix was generated to assess genetic relationship and among the genotypes. Then UPGMA software is used for cluster analysis. Then coming to result part, morphological characters recorded, 26 genotypes which include exotic, indigenous and wild species of cassava. The characters includes emerging leaf color, mature leaf color, petiole color, number of leaf lobes, young and mature stem color, stem branching, tuber skin color and ring color and flesh color shape. From that, observe that there is a wide variation observed for all the MO characters and roots are present in wild species. These are the table for characters, morphological characters. These are the selected some of the genotypes. Out of 10 SSR primer screening, 8 including SSR 6, 21, 32, 40, 42 and 77, NS 136198 revealed a polymorphic bands. A total of 21 polymorphic bands were scored and averaging 2 points bands per primer. These are the similarity index by using Shannon index similarity metrics. From that, the similarity coefficient among the cassava genotypes ranged from 0.29 to 0.95, indicating moderate to high genetic diversity. Released varieties or exotic varieties shows high similarity with each other, suggesting a narrow genetic base due to common ancestry or breeding history. Indigenous genotypes exhibited a moderate high similarity with the released varieties, indicating possible shared genetic background. While genotypes showed a low similarity values with the released indigenous genotypes, reflecting the high genetic divergence. From the dendrogram obtained, there are majorly 4 clusters. Cluster 1 include Albert, only one genotype. Cluster 2 comprise 18 genotypes including both CMD resistant and susceptible varieties. Cluster 3 group triploid and backrows with wild genotypes, for example Sri Aburva and Sri Adulya and BC wild. And cluster 4 contain a wild relative such as Manihod glasiovi and Manisod pseudoglasiovi. Interpretation: high similarity among released varieties suggests genetic uniformity, and indigenous varieties represent an intermediate genetic group, while relative possess unique alleles can be used to broaden the genetic base in cassava breeding programs. Clustering of released varieties suggests a narrow genetic base. Indigenous varieties act as a link between cultivated and wild genotype. Distinct clustering of wild genotype confirms their roles as a valuable source of novel alleles. Conclusion: the morphological and characterization shows the genetic diversity analysis between each other, and the study confirmed the extent of genetic diversity among cassava genotypes and successfully categorized on the molecular data. Then UPGMA analysis clearly separated the released indigenous and wild cassava genotypes, revealing a substantial genetic diversity and the importance of wild relatives of crop improvement. And it's also help in understanding the genetic relationship and can support breeding strategies, especially for disease resistant and genetic improvement. Thank you.
Thank you. The floor is now open to questions from the audience. Anyone?
So actually, this kind of studies are done, no, comparing many cassava varieties before also. So what was the uniqueness or difference in your work compared to the previous one?
Actually, ma'am, it's a preliminary work. We further do more, including genotypes, etc.
So is this a repetition of existing available information, or is there anything new that you?
There is a wild cassava. In wild cassava, the flowering is not happen most probably. So and also there is a triploid genotypes are released. So actually the flower morphology we have to confirm how the.
No, my question is, did you, when you the results that you showed, does any of the result have something unique or different from what was existingly known previously, or was that information which was, repetition, because cassava is a highly worked plant in your institute?
Third question is it the first type of work using SSR markers?
No.
It has been done before. How is it different from the previous work?
Actually, I don't know clearly that much.
So you said you tested tested a lot of the SSR markers and you picked 8 or
Yeah,
10. How many?
only 10 SSR markers are used.
Out of you tested 10,
10.
and out
Out
of
of
10
8.
you have used 8. 8 you
No,
found to be.
out of 10, 8 only give the polymorphic bands.
Okay. So is 8 a good number to do this kind of study?
According to gel, I think it's suitable, but my mentor says that we used more SSR markers for that.
More markers are
Yes,
needed,
yes.
right? So when you were testing these markers against all the genotypes,
Yes, ma'am.
did you find any difficulty because the polymorphic markers they tend to show the same when the backgrounds are similar. So you said your released varieties, they all kind of show low genetic diversity, right? What is the basis of that?
I don't know. Once.
Why would you think that the released varieties have low genetic diversity?
Because how can I say? You asking why?
Your research shows that all the released varieties, they have low genetic diversity, right? They tend to cluster together.
Yeah.
Why? Did you go back and check why? What was the reason?
No, ma'am. Actually, not.
But what do you think would be the reason?
It's already the means I think it's heterozygous in nature. So always the genetic makeup will be changed according to
No, they have low genetic diversity,
city.
meaning that they essentially have the same backgrounds, right?
Sorry, ma'am.
My feel was that the wild varieties generally flower and the cultivated ones may be reluctant, but you are given a
No,
opposite.
in wild varieties, the flowering is very less. Maybe and seed setting also very less.
Any reason for that?
We don't know. I think the fertility is not happening. In triploid plants, the anthers are very sterile. So when we do pollination, it's not giving result. It may be
The wild varieties are triploid.
yeah. Some Sreya Purva, Sreya Tulya we have. They are triploid.
Cultivated ones, what is the ploidy?
Diploid.
Anybody who would like to ask questions? So, because of the triploidy, is there a vigor increase in the wild variety? Vigor of the leaf or something like that?
The leaf was very thick.
Thick. What about the tubers?
Similar to wild plants.
What about the tubers?
Tubers are no yield is very less.
Less.
Okay, thank you.
Thank you, Ms. Nandisi. Next participant, Dr. Sangeeta Bishi.
Thank you.
Good evening to all. The topic for my presentation is identification of a novel source of sweet potato weevil resistance from wild Ipomia mauritiana. Myself, Sangeeta. I am working as senior scientist in ICR CTCR. Okay. Okay, this one for change. Coming to the introduction, sweet potato, we all know that is the fifth most imported crop, with China being the largest producer and Asia with the largest area of cultivation and largest area which contribute for sweet potato cultivation. And the crop is most important because of its nutritional value, like it's a major source of vitamin A, beta carotene, anthocyanin. And this is one of the crop which Janlo got the World Food Prize for the vitamin biofortification in orange flower sweet potato. It is a major source of vitamin A deficiency, for the alleviation of vitamin A deficiency, which is used in Africa. So coming to the problem, as we all know, sweet potato, the major pest is sweet potato weevil. Actually, it is a major pest, a noxious pest with about 20 to 55% average yield loss, and severe infestation causes 100% loss. And how this happens is, the biology is the plant itself, that is the sweet potato itself, will release some pheromones or semiochemicals which will attract the sweet potato weevil, so that the weevil comes, lay eggs on the tubers, and it will lead to whole yield loss. So with respect to this, right, we don't have a resistant source till now. So we started the screening of wild Ipomia species, and from our institute we reported Ipomia mauritiana resistant to sweet potato weevil. That is a wild Ipomia species which is tuberous, which is flowering with the chromosome of diploid 2n is 2030. And the leaf protein, that is the crude leaf proteins, we have tested against a weevil that is having insecticidal activity. Then with respect to this, we have selected Ipomia mauritiana as a resistant source and Ipomia betatus, that is our sweet potato, as a susceptible source. Contrasting phenotypes, genotypes we have selected, and we have done the transcriptome sequencing, and we have done the metabolomics, that is a whole integrated approach to know the resistant source and to know what is the biology behind the resistant source and how it can be utilized for developing host plant resistance in sweet potato. So the objectives were to identify the candidate gene responsible for sweet potato weevil resistance, to elucidate the genetic pathway responsible for sweet potato weevil resistance, and to identify the bioactive compounds. Already I said that the metabolomic approach. So both we have combined transcriptomics and metabolomic approaches to find a resistant source and for developing a resistant source in sweet potato. Because this is the first study, actually in 2022 from China, this is only report for sweet potato weevil resistant. That is 2 genes they have reported. That is sweet potato weevil resistant 1 and weevil resistant 2 gene. That is in chromosome 9 and chromosome 7. This is the only report for sweet potato weevil resistance. So we have done the transcriptome. Then we have elucidated those 2 genes, that is WRKY and DHQ, that is dehydroquinate synthase gene. These 2 genes were the ones which were reported from that already reported variety. So we have selected our Srivardhini CTCRI Indian cultivar with respect to Ipomia mauritiana. We have taken the full length sequence of 2 genes from that Ipomia betatus reference genome of China. Then we have compared, we have done the blast analysis, and from the transcriptome also we have taken these 2 genes. We have compared, designed the primers with the PCR amplification and sequencing. We would found that first gene, that is the WRKY gene, is present in our Indian cultivar. That WRKY should bind and it should activate that W box element. It should activate the second gene, that is DHQS. But in our Indian cultivar, that is in Srivardhini, that is one of our Indian cultivar, that it is absent. So that is one of the reason for the susceptibility of Indian cultivar sweet potato. So now we have come to the conclusion that this gene is not present. Actually it is present, but in the form of inactive receptor like kinase in our Indian cultivar variety. So now we have started with these gene and these primers. We have started screening of our CTCRI varieties and our sweet potato germplasm for a resistant source. Once this is present, but it is in the form of receptor, in a inactive receptor like kinase, and we would get only about 350 base per gene sequence. So from this is the first part. Then the rest of sequence analysis we are doing with the Ipomia mauritiana and Ipomia betatus. Then this is the heat map of those 2 genotypes, and these are the differentially expressed gene. That is the first set we have selected for sequencing. We have selected for primer designing and RTQPSR study. Then coming to the elucidation of the genetic pathway, we could found that the alkaloid tyrosine metabolism, that is the secondary metabolite present in Ipomia mauritiana, is responsible for its resistance. That is the insecticidal compounds is responsible. So with respect to this pathway analysis, we have gone for the transcriptome. This is the SRA and TSA submission of transcriptome sequences of both Hypomia mauritiana and Hypomia batatas, which is submitted in NCBI database. Then coming to the identification of bioactive compounds, we have done the sweet dry powder of Hypomia batatas and Hypomia mauritiana we have selected. We have done the HPLC analysis of the phenolic compound, which is with respect to insecticidal activity. And we would found that in Hypomia mauritiana, caffeic acid, gallic acid, actually these are the ones which is related to shikimate pathway, which is the one which is reported that shikimate pathway and the metabolites accumulation of in the shikimate pathway will give resistance to sweet potato weevil in Hypomia batatas. But in our Indian cultivar that is absent, that is the accumulation of that metabolite is absent because the shikimate pathway is not activated because of absence of the second gene. So with respect to this, we have come to the conclusion, these are the HPLC chromatogram of those compounds. And this, with respect to this, we have come to the conclusion that this transcription factor related to sweet potato weevil was identified in Indian cultivars, but the second gene was absent. It is in the form of inactive receptor-like kinase. So we have screening our other Indian cultivars from sweet potato germplasm, and this is the first study with respect to wild Hypomia mauritiana we have done. Then with respect to comparative transcriptomics, we have found that the genetic pathway analysis, that the flavonoid biosynthetic and selenoid tyrosine metabolite pathway is responsible for the weevil resistance. Then coming to the metabolite, the phenolic compound accumulation with insecticidal activity was in Hypomia mauritiana, which is responsible for the insecticidal activity. So this is one of the first report from our CTCRI, and we are screening and we are doing other work. Once this gene has been identified on this second gene, we will be initiating a genome editing approach for developing sweet potato weevil resistant varieties in sweet potato. These are the references, whatever I have used in the paper presentation. Thank you.
Thank you, Dr. Sangeeta. Any questions from the audience? Scholars may please ask. Okay, there is one scholar. Please, can you hand over the mic?
Okay.
As you have already mentioned that this weevils is a greater problem for sweet potato. Right now, how it is means overcoming this problem?
Actually, overcoming the problem means we have some of the varieties like early maturation variety. Whatever sweet potato we are cultivating, it is getting infested with the sweet potato weevil. With maybe some 50% of the population or some tubers we will get without infestation. Like that, we are maintaining. It is a big problem for this crop.
Is there any other remedies?
And then some cultural practices we are doing. Then imidacloprid spray, it is there. Then some biocontrol agents, they are saying like Metarhizium vivaria. But still, the weevil population will be there and it will cause up to 25 to 50% infestation. We are managing with the cultural practices, and some early maturation varieties will be there, like before 90 days. That is means it will skip, but that is season dependent variety. But still it is a problem.
As you have already stated that you have compared two species, right?
Wild and?
Yeah, wild one. So I think the wild one has less infestation compared to that.
Wild one is 100% free of infestation. That's why we selected that.
Okay. Is there any other phenolic compounds that is releasing that can prevent this weevil's population?
Actually, in this wild, we got cunic acid. We have estimated cunic acid. We found cunic acid from its analysis. But the same in that paper, whatever reported in 2020 with the sweet potato weevil resin, the same compound is reported from sweet potato in that paper. So the same compound in this paper, the same compound, this wild genotype is also emitting. Means wild genotype, it is there. So that is the major factor, cunic acid.
As you have said that this cunic acid is releasing, is there any technique that we can take out this cunic acid and make a component or make a
Actually, we are now testing it. We have purchased the quinic acid, that is pure, extra pure chemical, and now we are testing it against weevil.
As an outsource.
Outsource.
Okay,
Okay.
thank you.
Just for our study, and we have to screen, that's what.
Dr. Sangeeta, just to make myself clear, so
Okay.
Ipomia mauritiana is the resistant variety. So
Yeah.
you're comparing the resistant variety with a susceptible variety, and you did a transcriptome analysis. So, but I was curious that you went straight to that sweet potato resistance 1 and 2. Why did you go for that?
Ma'am, actually the first thing is for sweet potato, no resistant variety was reported, no resistant source was reported.
No,
So
you yourself said Ipomia
Ipomia
mauritiana.
mauritiana, ma'am, that is from our institute only we reported. So actually, up to the 2022, there was no resistant gene or resistant pathway like that. Nothing is reported. This is so. First, when we done the transcriptome, we have taken.
So
the
you did that work prior to the release of that paper, you mean to say?
Yeah, actually, we started this work and we got this genotype, wild genotype in 2020. And we were screening and we have done the transcriptome. In 2022, this is released. This paper is released. This is a Nature publication from China. Then why we have done straight is, ma'am, those two genes, they have reported. So from that reference, we have taken the full length gene from chromosome 7 and 9. And from this transcriptome also, just first, as a first step, we tried. If that gene is there, that second gene was also present, we can go for whether it is possible for a genetic transformation or gene editing study. For that only first we have done that.
But the second gene is missing in your case,
But in,
right?
in Sri Vardhini it is missing. Now we are screening other varieties
What
also.
about Ipomia mauritiana?
In Ipomia mauritiana also that is not. Those two genes are not there.
So that means it's either being activated by a different pathway,
Yeah,
or a
ma'am, it
different,
may be. That's
a
what it-
different gene is involved, right?
Yeah,
A
it
different
may be.
second gene.
It may be different pathway may be involved because in my mauritiana so many secondary metabolites are involved. Means accumulation is more compared to Ipomia patatus. So it may be due to some other pathway.
So why didn't you look at, when you compare the transcriptomics, why didn't you look at the genes which are differentially expressed
That is
between?
done, madam. That is done, and we are screening and we are designing. We have designed the primers and we are doing the RT-qPCR study.
So which is your candidate? Which from among this, what have you narrowed down to?
Actually, I have narrowed down to the genes which are conferred to secondary metabolic pathway. That is that quinic acid pathway genes only I have standardized, which is related to quinic acid phenol metabolic pathway only I have narrowed down.
How is your transcriptome data? Is it in sync with your metabolic data? I mean,
Yeah, ma'am. That's what those pathway, whatever I got, the same compound estimation also I have done with the HPAT analysis. That is correlated.
okay.
That's what we are. Now we are screening it one by one. Actually, our germplasm and varieties we had to screen and we had to see.
Okay. Thank you.
Okay. Ipomia mauritiana.
It's not edible, is it?
No, sir, it is used in medicinal property, but traditional.
This compound is overexpressed in the wild one. How do you make your cultivated one resistant if you are going for any process where you want to express this in the plant? That would make its quality,
No, sir, actually from those we are not taking any gene.
how do you plan to impart resistance?
Actually,
Go to your
for that part only, sir, we have we are first checking those two genes because that is already reported from sweet potato. That first two genes, SPWR1 and R2. If we got SPWR2 in any of our variety, we can go with that. No need means for that gene we have to activate second gene. First gene should bind the second gene promoter and it should activate. That is the genetic mechanism. And why this wild we have selected is, so far we are screening, even the wild Ipomia also we are not getting a resistant source. This is the only one we got resistant as a tuber variety from wild Ipomia.
what is the compound which is expressed on infection?
In this, it is in Ipomia mauritiana also cunic acid it is reported.
So is there any deterrent factor, in the sense that
Sir, in
I'm saying that when something is overexposed, for the resistant process, it may lose its edibility.
Sir, actually, in whatever the papers reported, it should not affect the flavonoid biosynthesis pathway. That is the sweet potato, that compound, everything that is in flavonoid biosynthetic pathway. So, whatever process, or if it is, we are going for a gene editing approach or genetic transformation approach, we had to off-target mechanism is it should not affect the flavonoid biosynthetic pathway.
All these resistant genes are present in all the varieties.
Yeah, first is present, sir. Second one only we are checking it because half the sequence only we are getting, like up to 350 to 500. The rest it is missing. So we are screening in our varieties. Not abs, it is present, but it is in the form of an inactive receptor kinase. Either we should be able to activate inactive receptor kinase gene is also the genes which are involved in plant defense mechanism which contribute to secondary metabolite. So that also we had to check. Then only. So ma'am, from blast analysis and from sequencing, we got it like from the transcriptome sequences, we got like it is in the second gene in our variety, it is going to the blast or similarity to inactive receptor kinase.
No, that doesn't mean that it is inactive, na?
Yeah, that's what we had to check. We had to screen and again we had to validate.
So when you check the expression of that particular gene, did you do that independently apart from the transcriptomics? Did you do an RT-PCR?
That now we are doing, madam, with RT-qPCR we are doing it. Okay.
You said that some particular gene which was present in the resistant variety was not activated in this susceptible one.
In our,
So my doubt is, did you check the genome whether this gene is there, or you are telling only based on transcriptome sequence?
No, we checked the gene.
It is there in the genome.
There, but it is inactive.
It is inactive.
That we checked first.
Okay, so it is inactive because did you find any reason? Is it due to some epigenetic silencing or something like that?
Ma'am, in first paper that whatever reported, they are saying like it is a epigenetic mechanism or due to the chromosomal addition deletion, it may happen.
So if it is chromosomal addition deletion, you can easily check
it
by sequencing.
Yeah,
So-
it is mentioned like that.
So you were telling about genome editing approach.
Okay.
So which genes would you want to target for that?
The same one, madam. This first one WRKY and the second one DHQS, because if that gene DHQS is activated, that doesn't affect the flavonoid biosynthetic, or it won't affect the nutritional composition of that sweet potato. That's what first we checking for that one. Otherwise, from our transcriptome, if it is not working, we have to select for the other genes.
So if it is a case of addition deletion, you can use genome editing.
My
What if it is a case of epigenetic modification? What will be your approach?
If it is an epigenetic approach, madam, either I had to go for a genetic transformation method, otherwise I had to trace again other genes. Other means in this differentially expressed DEGs, we have got about 400. From that, we have narrowed down.
So, what is your opinion about epigenome editing?
Epigenome editing, ma'am, so far we have in this crop, and again for this crop, another one challenge is that gold standard reference genome is not there in this sweet potato. This Taichung reference genome, that is Taichung variety China reference genome is available. Whatever from that we are standardizing, we are extracting the gene sequences and we are seeing. That is also a challenge.
Good work.
Okay, thank you.
Thank you, Dr. Sangita. Now, the oral presentation session on horticulture and forestry ends here. As we draw this session to a close, we thank our respected judges, Dr. Jeannie Anthony and Dr. Jasmine Shah, for their thoughtful contributions and support.